Good evening, my dear friends. I welcome you all to the Hindu Science and Technology Analysis on July 31st, 2022. Friends, all of us have heard about cancer, and we have all heard about chemotherapy. Chemotherapy is the one of the three kinds of therapies which are applied to cancer cells to treat cancer. The first is surgery, that is one kind of therapy, removing the cancer cells, the cancer affected, tumor affected portion. The second one is radioactive therapy. Under radioactive therapy, the radioactive radiation is being applied to the tumor affected cells and is used to eradicate the cancer cells. Chemotherapy, on the other hand, is the application of systematic medicines for the eradication of cancer. So, how this cancer is formed, my dear friends? It is a combination of the process which inhibits the signals not to be reached in NK cells. NK cells are naturally found in our blood. These are natural killer cells. Body often produces tumors of various kinds as we face the threat from a cancer developing. However, the natural killer cells get the signals from the cancer cells, and through this lytic process, this is called a lytic process, it goes to the cancer tumor cells and destroys the cells. The development of cancer is owing to this lytic process being breaking and stopped by the naturally induced cancer cells, which stop the signals to travel to the natural killer cells. Natural killer cells are always sleeping in our bloodstream. Unless motivated and induced by the signals from reaching from the cancer cells, these NK cells are always sleeping. And when the NK cells are getting this signal from the cancer cells, they go and patrol. That is, patrolling means in the night we observe that policemen are patrolling the streets and the roads of our locality. Similarly, the NK cells go on patrolling and finding out where these cancer cells are located and destroying the cancer cells there itself. This is the way in which the natural NK cells destroy cancer. And there is a process in which, a genetic process in which cancer develops in our body. The explanation is as follows. Friends, I hope you know how the cells divide and the older cells die out. There is a nucleus in our cell which produces the messenger RNA, which transcribes the gene's transcription, transcribes the cell division information into the messenger RNA, which travels to the ribosome, and the ribosome produces the synthesizes the proteins and produces the cell division and cell growth. In the nucleus, there is chromosome. The chromosome contains the genes. The genes are the form of a DNA, the double helix structure. This double helix structure contains the gene expressions, that is information with regard to the cell division and cell growth, and varieties of other information related to the characteristics of the color of the eyes, related to the characteristics of the particular cell. There are lots of different kinds of cells in our body. There are 66,000 billion cells in our body. Some are related to bones, some are related to blood vessels, some are related to tissue, some are related to the organs, some are related to the muscles. So different types of cells. So all this information, the gene expression is contained in the chromosomes, that is DNA, the double helix structure. And how this cell division is happening or the cell growth is happening is that from the double helix structure, the transcription happens to the messenger RNA. And this DNA, when it is coiling, it is unfolding, uncoiling or unfolding. Like the two ropes tied together, we try to pull it, stretch it, it uncoils. The two ropes bound together uncoil. Sometimes we do it with some nylon ropes we are having. Similarly, the DNA, the double helix structure also uncoils. And when this uncoiling is happening, there is an enzyme called the topoisomerase 1, which strikes the double helix structure of the DNA and cuts one of the helix. That is, one of the strand is being cut by the topoisomerase 1. And it expresses itself, instead of the normal DNA, it expresses itself into the DNA, and it relaxes the uncoiling of the DNA. That is, when the ropes are uncoiling, when we leave the ropes, it coils like a snake because it relaxes. We release the ropes, it relaxes, and then the uncoiling, like the uncoiling of the coiling of a snake is happening. In the same way, when topoisomerase 1 is hitting and it expresses itself in the normal DNA, what happens is that the uncoiling stops and the DNA rolls. The remaining DNA structure rolls. And this is the relaxing of the DNA. And the cancer is inhibited, is induced in this way. This faulty DNA is developing and it forms, grows, multiplies, and the multiplication is very fast. And this type of tumor is going on developing without any control. And it forms its own blood vessels. It's a very interesting point that from the amino acids taken as a food, it develops its own blood vessels and it survives on its own. And when it enters the normal bloodstream, it flows to the other parts of the body, and what happens is that the other parts of the body are also affected with this tumor. This is called mysteries. Mystitis. This is mystitis. That is affecting the other parts of the body. So, what is the anti-cancer treatment, that is chemotherapy, is working? The systematically induced medicines are going to control this topoisomerase 1. Hitherto, the medicines induced used to control this topoisomerase 1. However, it was later found that another villain is there. A drug called camptothecin was induced as part of chemotherapy to the target topoisomerase 1. This has been happening for a long time. Then what happened? Another enzyme called protein arginine methyltransferase 5 repaired this process and developed anti-chemotherapeutic resistance. So this protein arginine methyltransferase 5 resisted the chemotherapy. Hence, we tried to induce medications, chemotherapy directed at this protein arginine methyltransferase 1 also. This camptothecin was applied on both topoisomerase 1 and the protein arginine methyltransferase 5. Camptothecin was going on applied. However, after some time it was found that another villain is also there, which repairs the cells, which repairs the coiling. That is, the topoisomerase 1's process of destroying the DNA is going on. There is no control on that. The camptothecin is although working on the targets. Targets means the places where the medicine has to be or the chemotherapy has to be applied. Though it is going without disturbance, but after some time again this cell is recouping with the help of repair mechanism by this protein arginine methyltransferase 5. There was another villain which was helping this protein arginine methyltransferase 5 to diffuse the camptothecin, the medicine which we used to apply for chemotherapy. The name of another protein or another enzyme which induced these processes were hidden until now and we have now discovered. That is TDP1. This another villain is named TDP1. That is tyrosine DNA phosphodiesterase. Tyrosine DNA phosphodiesterase 1. This is responsible for blackmailing the other process camptothecin's application of topoisomerase 1 and protein arginine methyltransferase 5's activities. So this another enzyme, tyrosine DNA phosphodiesterase 1, had to be controlled. So what we have done is that we have done chemotherapy with the usual medicines and a little dose of camptothecin. Thereby we have tried to control the cancer cells, the growth of the cancer cells. The clinical trial phase 2 is completed for this chemotherapeutic medication and it will be soon out. Scientists deserve a very great applause from us for this discovery, my dear friends. Thank you. Thank you for your patient hearing. Meet you tomorrow with the Hindu and the Business Line editorial analysis. Till then, bye. Good night.